Hello students, welcome to SK Biology. In this video, we will understand packaging of DNA inside nucleus. We all know that now DNA is polynucleotide long molecule. Then the question is how DNA is packed inside tiny nucleus. We will get answer to this question in this video. Before going into details of packaging of DNA, I want you to know what exactly packaging of DNA means. We will understand this with this example of rope. If I tell you to fit this rope inside small circle, can you fit it inside as it is? No, it is not possible, right? Then if I tell you to fold this rope in a compact form and then try to fit it inside small circle, is it possible now? Yes, you can fit it easily inside small circle. The same thing happens in case of DNA packaging. This rope is nothing but polynucleotide DNA. Large oval shape is actually eukaryotic cell. Small circle is nucleus and packaged rope is actually packaged DNA. So, DNA packaging refers to the orderly and compact arrangement of long DNA molecule within limited space of cell. For example, length of human DNA is nearly 2.2 meter, but it is packed inside nucleus whose diameter is around 10 micrometer. This is possible due to highly organized packaging system. Let us understand how DNA is packaged inside nucleus. Firstly, we will understand DNA packaging in prokaryotic cell, that is bacteria. Bacteria does not contain true nucleus. DNA is compactly arranged in a region called nucleoid. Packaging is achieved by supercoiling, in which some associated proteins like HU protein helps it in packaging, forming compact supercoiled nucleosome structure as shown in figure. Now, let us understand DNA packaging in eukaryotic cell. In eukaryotic cell, it is more complex and highly organized due to presence of true nucleus. There are 4 packaging levels of DNA. First level is formation of nucleosome. Nucleosome is basic unit of DNA packaging, just like single unit of rope shown here. Second level is formation of chromatin fiber, also called as solenoid structure. It is like folding of rope to make it compact. Third level is formation of loop domain. It is similar to tying a knot in this rope, so that more compact structure we are getting. Fourth level is chromatin formation, which is like final compact packaged rope. We will understand this process of packaging step by step. This is double helix DNA. Size is around 2 nanometer. It is fully extended form of DNA. This DNA is wrapped around histone octamer. Octamer consists of 4 histone proteins H2A, H2B, H3 and H4. 2 molecules of each histone protein. These histones are basic proteins rich in lysine and histidine amino acid, which are positively charged amino acid. This positive charge on histone attracts negatively charged DNA. About 146 base pair of DNA is wrapped around histone octamer, and this octamer is connected to adjacent nucleosome by linker DNA, which is around 20 to 60 base pair long. Histone H1 binds to linker DNA and helps in stabilization. When number of histone octamer binds with each other it forms structure called beads on a string. The size of this structure is around 10 nanometer. This beads on a string structure coiled further to form 30 nanometer thick chromatin fiber stabilized by H1 histone, forming solenoid structure. 30 nanometer fiber forms looped domains. These loops are attached to non-histone scaffold. Each loop contains around 50K to 1 lakh base pairs. Loop formation is important for function and organization of genes. During cell division, this chromatin fiber becomes highly condensed. These looped domains fold and condense to form metaphase chromosome. Highly condensed section of this metaphasic chromosome shown here is measure around 700 nanometer. Complete compact metaphasic chromosome measures about 1400 nanometer in size, giving highly compact DNA. DNA packaging is not just to fit DNA inside nucleus. It also plays crucial role in cell, like gene regulation, DNA replication and repair, cell division, and it also protects DNA from damage.